Okay, so welcome everybody to the November Shanks Pathological Society Pathology Grand Rounds lecture. Today's lecture will be given by Professor Menna Clatworthy. Menna studied medicine at Cardiff. She completed her professional training in nephrology at Cambridge, having undertook a PhD at the University of Cambridge investigating antibody effector function in autoimmunity and infection. She was awarded the British Renal Association Raine Award and the Academy of Medical Sciences MRS Young Investigator Award. She completed a Wellcome Trust Intermediate Fellowship at Cambridge and at the NIH Bethesda. She's currently the Professor of Translational Immunology at the University of Cambridge Department of Medicine. She also works clinically as an honorary consultant in nephrology in Cambridge, and she has an associate faculty position in cellular genetics at the Wellcome Trust Sanger Institute. Her research is focused on understanding the regulation of antibody generation and effector function, novel methods of targeting humoral immunity in transplantation and autoimmunity, and the role of tissue environment in shaping resident immune cell activation and function, particularly in the kidney. She's an active member of the Human Cell Atlas project using single cell approaches, and this will form part of her lecture today, which is entitled Applying Single Cell Technologies in Nephropathology. Thank you, Menna, very much for speaking today. Thank you.
Thanks very much, Mark, for that introduction. Very, very kind of you, and also for the invitation to give this grand round today. Can I just check that you're able to see my screen and that you're able to hear me?
Yes, I can see you and see the screen.
Brilliant. Okay. So as mentioned, I'm going to tell you a little bit about our efforts to try and apply relatively cutting edge single cell technologies to human samples, so particularly in the kidney. And I'm a nephrologist. Actually, I'm principally working in transplantation now, but I guess when we're approached by any patient, so that's referred to us, whether that's a patient with a transplant or without, often they will be referred because their renal function is abnormal as measured by serum urea and creatinine. And of course, as a nephrologist, the key question that we want to address in such a patient is, well, why? Why is their kidney not working properly? And there are, of course, a variety of etiologies that can lead to an increase in creatinine and renal dysfunction. And those might include autoimmune diseases, toxins, infections, hypertension. And there are some very simple tests that we can do at the bedside that will help us to delineate that. So, for example, whether there's associated hypertension or an active urine dip. But ultimately, with many of these patients, what we need in order to gain the ground truth of what's going on in the kidney is a kidney biopsy. And this is something that we do relatively frequently, as you know, and it is, I guess, one of the reasons why I ended up doing nephrology, because when we have a patient, say, who presents with acute renal failure, we're able to do a biopsy in the morning, walk it over to pathology, and in the afternoon go back there and sit with the pathologist and see exactly what's wrong. So these are, of course, done under ultrasound guidance and a local anaesthetic, and we're able to get cores of tissue, and you're actually able to see at the bedside whether you've got kidney. You can even see the little glomeruli in the cortical samples, which is obviously what we're trying to get. And so I guess our assessment, and by our, I mean the biomedical community, so our assessment of renal pathology hasn't changed that much in several hundred years, in that what we principally do to make our diagnosis is to look by light microscopy and to look at appearances in the cell morphology of the intrinsic kidney cells, but also for the appearance of immune cells where perhaps we wouldn't expect them to be. So, for example, neutrophils in the context of infection, or in T-cell mediated rejection, we might see lymphocytes within tubules. And we are also able to go one step further. If we have sufficient sample, of course, we can look at ultrastructural components by EM, so For example, we're able to tell whether the foot processes are normal or not in podocytes. And we're able to also use immunofluorescence or immunostaining of some sort to identify different components of the immune system, like IgG, or evidence of complement activation, for example, C4D in antibody-mediated rejection. And one might see these as spatial biomarkers. And together, our renal biopsy then not only gives us the diagnosis by virtue of the features within each of those different modalities of looking at the biopsy, but also allows us to get an idea of prognosis. And by that, I mean, if you have a kidney that looks something like this, with a substantial tubular atrophy and interstitial fibrosis, we know, of course, that that's irreversible. And so that's very helpful in guiding whether we should or shouldn't treat a patient, and of course, in telling the patient their prognosis. But I want to emphasize, because it's relevant for the application of molecular technologies to kidney biopsies, that it's really critical in deciding what's abnormal to, of course, have a great grasp on what's normal. So just as normal reference is absolutely essential to interpret what's abnormal by light microscopy, the same will also be true in molecular pathology and in single cell molecular pathology, as I'll come to. So why might we move in nephropathology to a transcriptomic assessment of biopsy? So from these standard ways of assessing a biopsy, where we look at cell morphology, immune cell presence, some of these spatial biomarkers. So overall, these might give us perhaps, depending on what we've measured, maybe up to 50 parameters that we would use to define the diagnosis and perhaps the pathotype within that diagnosis. Well, even if we use something like bulk RNA sequencing or microarray, as was previously applied, well, this of course gives us many more parameters or pieces of information about our biopsy. So typically for bulk RNA sequencing, 20 to 30,000 parameters. And so this so-called molecular microscope approach has been applied to transplant biopsies in particular. And so collections of genes that are up and down regulated, say in T-cell mediated rejection versus antibody mediated rejection, can help to improve diagnostic certainty. And when I'm actually largely when I'm giving talks to clinicians in transplantation, I liken it to this. If you have a limited number of classifiers, so information, pieces of information that might classify these two objects, you might conclude that they were identical in their functionality and identity. But actually, if you get more information, you might conclude that you would prefer to drive the second object. So what we've been working on more recently is going even one step further. So not just thinking about bulk RNA sequencing, but improving resolution by thinking about things at a single cell level. And so the advantage of this is, of course, if we take a biopsy of kidney and we do bulk RNA sequencing and we identify, say, a bunch of genes that are upregulated and downregulated, what we don't know is the cell types. There are, of course, hundreds, thousands of cells within this biopsy. We don't know which of those cells and which of those cell types is responsible for one of these highly upregulated or downregulated transcripts. So we only know the whole. But single cell genomics allows us to get that information. So if we make single cell suspensions of our biopsy, we are able to profile the transcripts, the RNA transcripts within individual cells. And then we can cluster cells by virtue of similarities and differences in gene expression. And we frequently can project this information in 2 dimensions, as shown here. So where cells that are transcriptionally similar are clustered together. And then we can use our knowledge of canonical gene expression or markers that are associated with a particular cell type. So for example, macrophages might express CD68 or C1q we also see, but in that way we can label each of these clusters. And the cool thing about this way of approaching the cell types that are present within any given biopsy and their transcriptional profile is that it doesn't require any prior information. So they cluster based on similarities and differences in gene expression, and we don't feed in any information to say, well, they need to express this gene or that gene. So what has allowed this? Well, what has really enabled us to apply this with higher throughput have basically been a number of technological advances. So there are two ways really you can do single cell RNA sequencing currently. And those are basically, you either generate single cell suspensions or single nucleus suspensions, and you use a droplet based platform. So this is where you force single cells and then some barcoded beads. So each of these beads has a unique oligonucleotide barcode. And then you put these or force these using a microfluidic system into an oil droplet so that every cell is associated with a unique barcoded bead. And then you're able to associate the transcripts within that droplet with that particular barcode. And so then you can say exactly what each individual cell is expressing. And the other way of doing this, and I should say there are a bunch of platforms that allow you to do this. We've used the 10x genomics platform, which I think is relatively commonly used, but there are other ones too. Now, the alternative strategy is to sort individual cells into wells of a plate and then sequence those individual wells. And again, there are ways of doing this at higher throughput, but generally speaking, although this gives you better depth of sequencing, it's more expensive. So it's these droplet-based platforms that have been applied. And so we use this technology to generate what I'm calling version 1 of a single cell kidney cell atlas. And to do this, first of all, you of course need a source of tissue. And here we had kidney samples from across lifespan, including some prenatal kidneys. And these were obtained from the Human Developmental Biology resource, which is based both at Newcastle and UCL. And we got these from Newcastle. So we had 6 prenatal kidneys from terminations of different sorts from week 7 to 16. And in these, we actually sorted immune cells, so CD45 positive and then CD45 negative cells. And then we had a bunch of postnatal samples. So we had some neonatal kidneys, and these were from children who were undergoing nephrectomies because of Wilms tumor. And these were taken from adjacent normal tissue. We had teenage kidneys and then a bunch of older kidneys. And these were taken from 2 sources. These were either the adjacent normal of tumor nephrectomies, or these were kidneys that were donated for transplantation in the UK organ donation system, but couldn't be used for a whole variety of reasons. So we generated single cell suspensions and then fed them into the 10x system. And we actually got a reasonable coverage of cells. So in the developing kidney, we obtained data on nearly 30,000 cells. And we've colored these different clusters according to the broad cell types. So I'll remind you that the kidneys develop when the ureteric bud invades the cap mesenchyme, and that interaction results in the formation of the renal vesicle, which is transformed to an epithelial cell type, forms the S-shaped body, and then ultimately the components of the fetal nephron, so things we recognize like podocytes, proximal tubular cells, etc. And so you can see in these green clusters, we were able to identify cap mesenchyme cluster through to these terminally differentiated cell types like podocytes, proximal tubular cells, etc. There was also quite a lot of stromal cells, some endothelial cells, and most interestingly to me, so my lab is mainly focused on immune cells and tissue immunity, as Mark told you, and so we were interested to see that there were a whole bunch of immune cells within the kidney. And these included macrophages, which, as I'll come to, we were expecting, but also some adaptive immune cells like B cells and T cells. And in fact, because we had samples from different points in fetal development, we were able to plot the appearance of these different cell clusters over developmental time. And this is shown here. So at the start, we could see early on in terms of immune cells, we find macrophages, but later on, some of these adaptive immune cell types appearing. So then in the adult kidneys, or the postnatal kidneys, we generated data on 40,000 cells. And again, you can see we had information on the nephron epithelial cells of different sorts. You can see a whole lot of this was proximal tubular cells. But again, we had a smaller amount of stromal cells, some endothelial cells. And again, perhaps to our surprise, because, bear in mind, these are normal kidneys. These individuals don't have kidney dysfunction or kidney, or at least overt kidney disease. So we could still see a whole bunch of immune cells, so including mononuclear phagocytes, but again, B cells, T cells, and NK cells. And the great thing is about this data, and I'm not expecting you to read this in detail, but with each of these cell clusters, we're able to define the genes that are most upregulated or most highly expressed within those clusters. So we were able to make a transcriptional atlas or map of the entire cellular landscape of the human kidney, and both in development and in adulthood. And that is useful in a whole variety of ways. And particularly I would emphasize to define what's normal, okay? And so again, we need a normal reference data set if we're going to start to use these technologies to begin to better define pathology. And one of the questions, or one of the areas that we used this to address, was really a long-standing interest of my lab, which is, what are the tissue-resident immune cells that live in human kidney? And the historic view of this has really been that this is limited in health, at least, to mononuclear phagocytes, so that is macrophages and dendritic cells. And that largely other cell types originate or are activated in secondary lymphoid organs like the spleen and then circulate, and then perhaps they may be called to tissues like the kidney, so some neutrophils here only during infection. And we know and I should say, we know this principally from mouse studies where you can do fate mapping experiments, that macrophages that live within all organs and within the kidney in particular have two sources. They can either be seeded prenatally from yolk sac or fetal liver precursors, or later in life, postnatally, they can come from hematopoietic stem cells via monocytes. So there are two sources. But all of that, certainly when we first came to this, which is now maybe 5 or 6 years ago, all of that was largely defined using studies of mouse kidneys. So to bring this through to human, what we first did, well, the very first thing I did when I set up my own lab was to get permission to take kidneys that had been donated for transplant but couldn't be used. So we could actually have large kidney samples that we could begin to profile. And what we first did was just high dimensional flow cytometry to look for what mononuclear phagocytes were present in human kidney in health or in homeostasis. So we were able to identify CD45 positive, so immune cells in these cell suspensions that were MHC class 2 positive, CD11C positive. And then we could use some classical MMP markers like CD1A and CD14 to define two major subsets, though. So there are CD14 positive mononuclear phagocytes, and CD14 negative. And you can see that we found, interestingly, there was a difference between the frequency of these in the cortex of the kidney, so the outer bit, versus the medulla, and that the CD14 positives were more frequent in the medulla. And we went on to look at, well, if that was the case, were there differences in the expression of chemokines? And these are, of course, molecules that determine where immune cells position themselves. So using bulk RNA sequencing data, we asked whether chemokines were differentially expressed between chunks of cortex and chunks of medulla. And you can see, so here, highly expressed chemokines are in yellow and lowly expressed in blue, that there are a whole bunch of chemokines that are more highly expressed in the medulla versus the cortex. And two of those include CCL2 and CX3CL1. So which are chemokines which, based again on mouse work, would be predicted to be able to attract mononuclear phagocytes. And so I'm not going to go through the details of this study because we published this a little while back, but basically we showed, using a combination of human kidney samples and bulk RNA sequencing and immunofluorescence staining and mouse models, that actually it was the high sodium concentration within the medulla of the kidney that led to epithelial cells to produce CCL2 that could potentially attract monocyte-derived macrophages and actually position them in the medulla where they were ready to attack ascending infection. But this, our early studies here identified some limitations with the approaches of both using flow cytometry, immunofluorescence staining, and also bulk RNA sequencing, in that you are trying to find, say, cells based on your prior knowledge of markers, which may or may not be true or helpful. It doesn't allow you to approach what cellular heterogeneity there is in an unbiased way. It also doesn't really allow you to understand much about the origin of cells. So that question that's there from mouse models, are these cells originating from yolk sac or fetal liver precursors versus monocyte-derived mononuclear phagocytes, for example? And it doesn't allow an unbiased assessment of all cells that are capable of responding to immune perturbations. And that might not just include the resident immune cells, but those that come in, but also even potentially the non-immune cells that live in the kidney. So if we then take, those difficulties or limitations and say, well, could our version 1 of the human kidney cell atlas help to address some of those problems? So I'll show you, some of the analyses we were able to do. So first of all, if we zoom in now, I'm not showing you all of the clusters. So this is removing all the epithelial cells, stromal cell, endothelial cells, and now just doing some higher resolution clustering on the immune cells, and in particular here in this right-hand UMAP, the mononuclear phagocytes. So actually, we could identify 4 different clusters of mononuclear phagocytes, which we labeled A to D. And based on marker expression and some other analyses, which I'll show you, we identified MMP-A and B as being monocyte-derived macrophages, likely, MMP-C as being dendritic cells. You can see MMP-C highly express MHC class 2, and some other DC markers. So, for example, CD1C, XCR1. And then MMPD we labeled as tissue resident macrophages, and I'll show you the data on that. Now, interestingly, going back to our previous FACS studies, I emphasized to you one of the markers we used was CD14. Well, that was actually highly expressed in 2 subsets, so MMPA and MMPD. So if we were just approaching this by FACS, we would have put these 2 together. But as I'll show you, they're extremely functionally different. So we were also able to look at the fact, and I emphasized to you that to our surprise, there were a lot of lymphocytes within these healthy kidneys, and that was in both fetal and in mature kidneys. And these expressed a number of markers that have been associated with tissue residency in the mouse. So, for example, CD69. So suggesting that, in fact, the kidney has a rich network of resident immune cells, both adaptive and innate, even in homeostasis. So going to these mononuclear phagocytes in more detail, I said to you one question that was outstanding from the mouse models was, well, are these macrophages in particular of two developmental origins? And might that tell us something about their function and ultimately their contribution to disease? So because we had data from fetal kidney macrophages and postnatal kidney macrophages, we could ask, are any of the mononuclear phagocyte subsets in the mature kidney, postnatal kidney, transcriptionally similar to fetal macrophages? And you can see that, so transcriptional similarity is indicated here by a lighter color. So really, the only adult subset that had any transcriptional similarity is this MNPD. So you can see this is transcriptionally similar to the macrophage 1 subset within the fetal kidney. And in fact, our MMPA and B, they're transcriptionally similar to fetal monocytes that were present in the kidney there. So this provides the strongest evidence to date that there are in human kidneys macrophages that and well, are these, does that matter? Are these the same transcriptionally? Well, if we do gene set enrichment analysis, so this is where we take some curated gene sets that are associated with different functions. So for example, genes that are associated with defense response to bacteria or phagocytosis. Sets are enriched within these different mononuclear phagocyte subsets. You can see that those that are enriched within MNP A and B, our monocyte-derived macrophages, are very different from those that are enriched within MMPD, our tissue-resident MACs. So these are very, innate defense response to bacteria, whereas these actually, there's a lot of complement genes enriched here, and as we'd expect in DCs, antigen processing and presentation. So based on these predictions from the transcriptomic data, we went on to do some functional studies. So what we did was to take single cell suspensions from human kidneys, and we incubated them with fluorescently labeled uropathogenic E. coli. So, the commonest cause of pyelonephritis, so a very relevant challenge for these macrophages in the kidney. And you can see if we then look at uptake, so phagocytosis of MNPAs, so and we identified a marker for those based on the single-cell gene expression as CD36, and MNPDs, so CD206, and that the MNPAs, as our transcriptional data would predict, are far more phagocytic than the MNPDs. So we were also able to start to address this question, well, what's the immune functionality in the kidney beyond those resident immune cells? So, or put another way, for example, do the epithelial cells have immune functionality? And, therefore, might they contribute to defense against infection, one example, or even have the capability to therefore contribute to immune-mediated diseases in the kidney? So one of the things we did was, again, gene set enrichment. This is actually just using Go terms, which is a different curated database, but same principles. So here I've got the epithelial cell clusters laid out from proximal to distal, so podocytes, proximal tubular cells through to pelvic epithelium. And what you can see, so again, lighter color is higher expression, is that in the pelvic epithelium, a whole bunch of innate immune response antimicrobial pathways are enriched. And in particular, if we then look at some of the leading edge genes within these pathways, these are a whole bunch of antimicrobial peptides that the pelvic epithelium is secreting. And this is even, this is again in homeostasis. So it's setting up a defense zone in homeostasis. So making a potentially significant contribution to kidney defense. And interestingly, if we compare the fetal nephron component, so again from proximal podocytes to distal pelvic epithelium, we don't see the presence or the expression of many of these pathways or genes. So suggesting that the expression of these antibacterial molecules, these antimicrobial peptides, is something that's postnatally acquired by the pelvic epithelium. And one could speculate that it might be due to, for example, microbial colonization of the gut. So the other thing we could ask was, well, what are the cell-cell interactions in the kidney? And we can do this by, again, in an unbiased way, saying, what are all of the receptors that are expressed by one cell type, say immune cells, and then what are all the ligands that are expressed by another cell type, say, for example, epithelial cells. So we can move from this situation, where I know that there are some chemokines that are more highly expressed in the medulla, but I have no idea which cell type is producing them, to this situation where I'm able to look at every single cell type, so podocyte, again, through to pelvic epithelium, and I'm able to say, what are the ligands, so here the chemokines they're expressing, and of all of my immune cells in this specific data set, so in the kidney data set, who is expressing the receptors. So, there's a bunch of interesting things here, some of which we're currently exploring. So, for example, podocytes express a lot of CXCL12 with the potential to attract immune cells that are expressing CXCR3 and 4. But what really caught our eye was down in the pelvic epithelium here. There was expression of a whole range of chemokines, so CXCL8, CXCL1, CXCL2. So these are archetypal neutrophil recruiting chemokines. So CXCL8 is also known as IL8. So this would predict that the pelvic epithelium is not only in homeostasis producing antimicrobial peptides, but also cues for the basal recruitment of neutrophils. And again, if we compared expression in the fetal nephron with the adult nephron, this expression of neutrophil recruiting chemokines was something that was only present postnatally. And we went on to confirm this a different way. So other than our single-cell RNA-seq data, what we did here was to take chunks of kidney, in the case of human, so either cortex or medulla pelvis samples. And we did qPCR for CXCL1 and CXCL8. And you can see that in homeostasis, within the medulla pelvis sample, there is more CXCL8. And then we also did a challenge experiment where we then incubated these chunks of kidney with uropathogenic E. coli. And you can see in this context, both cortex and medulla upregulate their CXCL1 and CXCL8, but it's particularly highly expressed in the medulla pelvis samples. And in the mouse, we actually have a mouse model of pyelonephritis where we catheterize a mouse and challenge sequentially or repetitively with uropathogenic E. coli. And we could see a similar picture. You can then dissect out the cortex and medulla and the cortex and then the medulla and pelvis parts. So again, mice don't have CXCL8, their neutrophil recruiting chemokines are CXCL1 and CXCL2, but you can see a similar picture. So in homeostasis, more CXCL1 and CXCL2 in the medulla pelvis samples, and a substantial increase when we induce pyelonephritis. And we use some immunofluorescence staining to confirm both the expression of antimicrobial peptides, so particularly lipocalin-2 here, so right at the border here in the pelvic epithelium. So this is the urinary space here, but also of CXCL8, so in exactly the same cells. And then again, moving back to the mouse model, we have a mouse that is a fluorescent reporter for lyse-M, which is highly expressed in neutrophils, so it allows you to track them. And you can see post-UPEC in comparison to PBS-challenged animals that these chemokines are very effective at recruiting neutrophils. So this, again, is the urinary space, and here is the border. So overall, that study, the generation of version 1 of the Human Kidney Cell Atlas, showed us that, actually in homeostasis, there are resident immune cells within the human kidney, that including innate and adaptive immune cells, that those include monocyte-derived and also tissue-resident macrophages that have very distinct functionality. That the epithelial compartment actually has an expression profile that suggests important immune functionality, including, for example, in the pelvic epithelium, direct secretion of antimicrobial peptides, but also of neutral recruiting chemokines. And this gives us, I think, an important reference or ground truth of the single cell landscape of the human kidney that we can then begin to use to compare pathological situations to. And so we've made all of this data publicly available, and this is actually in a browsable format. So even if you have no bioinformatic knowledge, you can say type in your gene of interest and have a look exactly which cell type this is expressed in at kidneycellatlas.org. So what we've been doing to build on this is to begin to incorporate a spatial element to this. So we've done some experiments now using spatial transcriptomics. So what this does is, we've used, particularly the platform we've used is the Visium platform. So this has within 50 micron dots. So it's not quite single cell resolution. So within each 50 micron dot on a slide, there is a barcode that you then put your section onto the slide. So any transcripts that are detected within that particular 50 micron area, you can assign to that space. And so we've begun to do this, and using this, we can identify different cell types based on genes that are highly expressed within these different spatial regions. And broadly speaking, you can see we can identify here, there's a lot of, say, glomerular components in the outside, and then more sort of medullary pelvic components on the inside. And we're working on building this app so that we have spatial data to go with our single cell data. And then the other thing we've been thinking about is how one could use single cell atlas data, and I'll just give you a few examples, and I may sort of speed up a bit here, but one way is, for example, I presented to you at the beginning that molecular pathology at a bulk level has that's been started to apply to transplant pathology, such that the presence of, for example, so-called endothelial cell transcripts is now part of the Banff classification for antibody-mediated rejection if your laboratory has that capability available. But this study was from Ben Humphreys, and what they did to start with was just do single cell RNA sequencing. This was just on one biopsy. So this is a transplant biopsy with mixed rejection, so both T-cell mediated rejection and antibody mediated rejection. So you could see they could find a bunch of immune cells, and actually, because they didn't have ground truth there, they assumed that all of these immune cells were pathological, but what I've shown you, they're not necessarily. But anyhow, one of the interesting things that they did was to say, well, some of those signatures that have been identified, including so-called endothelial cell transcripts, TCMR, ABMR, we can now say which cell types are these specifically expressed in, so we can begin to get much better resolution of the cells that are driving both our, I suppose, diagnostic features, but also, it gives us an insight into underlying etiology. So, for example, so-called endothelial cell transcripts, which are these ones here, so the genes are here, actually, there's a really small part of them, so about a third of them, that are endothelial cell specific, and the rest are expressed in a whole range of different immune cells shown here, including, for example, proximal tubular cells. So it really allows us to refine our understanding. The other thing it allows us to do, and again, this is so one example that we published in a review article. So if you think about genome-wide association hits, so these are polymorphisms that are present in the general population that might confer susceptibility to a trait. So in this case, to chronic kidney disease or decline in GFR. So there's a whole bunch of genes that have been identified, but now we can say which specific cell types in the kidney express these different GWAS hits. And again, that begins to take you to, well, what's pathological mechanism? So allows you to take the next step as to maybe which cell type you should be studying to work out why those polymorphisms ultimately lead to disease. And then the other thing, so this is the last thing I'll show you, is that we can also begin having defined ground truth to build disease cell atlases. And I'll just show you some data that we've got where we've been thinking a bit about lupus nephritis. And so I'll just remind you that, systemic lupus erythematosus is the archetypal multisystem autoimmune disease characterized by anti-nuclear antibodies and the deposition of IgG immune complexes within, for example, the kidney, causing a proliferative GN. And we also find aggregates of immune cells in some patients with lupus nephritis, and in mouse models, these have been shown to even be able to generate some of the autoantibodies. So we first applied single-cell RNA sequencing to a mouse model of lupus nephritis. So this is the MRL-LPR mouse, which has a mutation in FAS, which means lots of autoreactive lymphocytes live, and they get a lupus-like disease by 3 to 6 months of age, with lupus nephritis by 6 months of age. So you can see if we look at their kidneys and cortex to medulla, here I'm looking at the different mononuclear phagocyte subsets, you can see in the LPR mouse things become hugely deranged, and there are many more mononuclear phagocytes, so shown here by this marker F480 and also by 11B. And so what we did was to sort those mononuclear phagocytes from control and LPR mice, and we did single-cell RNA sequencing, and we could identify 2 different subsets, so 2 broad clusters, group 1 and group 2. And we could relate these back to our to markers, so 11B and F480 that we used by flow cytometry, and we could also relate these back to the origin of these mononuclear phagocytes by looking at signatures from yolk sac-derived macrophages, and those are enriched in group 1, whereas monocyte-derived macrophages were enriched in group 2. And we could see that some of these were much more frequent. So, for example, 1.4 and 1.5 in the lupus, and 2.3 in the lupus kidneys. And then we use spatial transcriptomics, and I think this starts to illustrate the potential power of this. And if you think about if we could apply this to human kidneys. So this is a cross-section of a mouse kidney where we're profiling within these 50 micron blocks the expression of genes. So for example, we can pick out archetypal glomerular genes, or proximal tubular genes, or pelvic genes. And so we can then plot out exactly where each of these mononuclear phagocyte subsets lives in the kidney, based on this gene expression. And we can also look for enrichment of different gene sets. So for example, genes that are associated with IgG immune complex stimulation, and find specific subsets. So for example, 1.4 and 2.3 that are highly enriched for this. We can map that spatially, again, going back to our spatial data. And we can also look for novel interactions between different cell types, showing that some of these resident mononuclear phagocytes produce some of the chemokines, so particularly CCL8, that would allow monocyte-derived macrophages to be attracted. And then we were also able to look whether any of these mononuclear phagocytes were producing cytokines that could support those B cell aggregates that I talked about, so particularly the B cell cytokine BAFF. And we found that in the LPR, a high expression of BAFF across some of these subsets, and it co-localized, particularly with B cell transcripts. But then what we can do is then go back to human data. So we took our data, so that is ground truth, and we took data from Arazi and colleagues that was published last year that had tried to profile the biopsies of patients with lupus nephritis. And the thing is with these biopsies, because they were clinical biopsies, although they had quite a few patients, the number of macrophages they had was relatively small. So these are the red cells. So they weren't actually able to define any specific clusters. But because we embedded this with our data, where we had a lot more cells, we were now able to divide up the lupus macrophages into monocyte-derived and tissue-resident and begin to ask, well, is it, as we see in the mouse, the tissue-resident that are producing BAF? And the answer is yes, and a bunch of other questions. So hopefully that second part has illustrated to you the way in which we could potentially use single-cell RNA sequencing data, for example, to profile abnormalities of tissue-resident mononuclear phagocytes in a disease like lupus nephritis, the way we can use a mouse model to discover features that may be pathological, but then go back to human biopsy data to actually confirm the presence of those and confirm their clinical significance. So overall, then I'll wrap up there. Hopefully I've shown you that single-cell RNA sequencing could be a really powerful tool for nephropathology. It's helped us define the landscape, the cellular landscape of the kidney in a more unbiased way, to identify unexpected cell-cell interactions, for example, the power of epithelial cells in controlling immune cell position, even in homeostasis, and also provides a reference data set that we can use to compare disease. So I'll just acknowledge the people that were involved. So with the Kidney Cell Atlas, so the two people that drove the analysis of that were Ben Stewart, a PhD student in the lab, and John Ferdinand, a postdoc, as well as a whole bunch of people who were involved in obtaining and processing the tissues, which is really not trivial. And then our collaborators, so Sarah Teichman at the Wellcome Sanger Institute, and Muz Hanifa at Newcastle for the Human Developmental Bioresource stuff, and our funders. And of course, really specifically the donors who consented to allow their tissues to be used for research, and the patients that allowed the excess sample to be used. And then for the lupus nephritis, again, Ben and John, but also Georgie, Nathan and Kelvin, and our collaborators at NIH, Richard and Mariana. And then finally, if I could just give one ad, if there's an enthusiastic, computationally leaning postdoctoral fellow, we actually have a job for you, which is between the Sanger and my lab in the LMB, closing date 30th of November. So if you want to get involved in this exciting area of tissue-resident immunity, then apply. And I'll stop there.
Okay, thank you very much indeed, Menna. That was a really fantastic talk, and you really showcased the power of the combination of both single-cell RNA-seq studies with spatial transcriptomic studies. You've shown how this can be used for a very powerful analysis for gene expression changes in disease processes. Can I encourage the audience to start putting their questions into the Q&A box, please, and we will answer these. So I see some questions have already started to arrive, so I'll start at the top and work down. So Amy Bowes asks, for your future work, do you plan to implement any joint genomics and transcriptomics single-cell studies? For example, copy number changes may be related to alpha-synuclein expression and accumulation in Parkinson's disease. If yes, do you have a renal disease type in mind that might benefit from such a study?
Yeah, so I think that ultimately the future of this will be in trying to integrate genomic data. So by that, I guess Amy's referring to, for example, SNP information, right? So polymorphisms that confer susceptibility, or copy number variants that confer susceptibility to diseases. And you know, that could be, there are a whole range of those. So for example, I mean, some of the things that I showed you there from the GWAS is for hypertension or chronic kidney disease. But I guess what's limiting our ability to do that at the moment is what you need for those studies to look at the effects of SNPs is that you need large numbers of patients. And at the moment, this technology is pretty expensive, right? So applying this across, population, so hundreds or thousands of samples is difficult. But I think as time goes along, the cost will come down, and we will be able to start to apply those. And ultimately, I think the way to apply it will be not just integrating genomic information, but also potentially epigenetic information as well. So there are platforms for doing single cell ATAC-seq too. So then we'd be able to think about how the chromatin landscape has changed and whether that might also inform, again, pathogenesis. So I think, yeah, all of that's for the future.
Okay, thank you. So I'll combine the next two questions, if I may. So
Yeah.
Stephen Notley asks, have you any plans to use your techniques on other pathologies such as IgA nephropathy and FSGN? And Simon Harrington asks a similar question about investigation of FSGS and its recurrence in transplant kidneys.
Yeah, so I think, like I said, we're at the beginning of trying to use these technologies and applying them to different diseases. And there has actually been one paper that has, or one study that has done some single cell RNA sequencing on IgA nephropathy kidneys. But I think the number of cells there was pretty small, and the number of immune cells small, and maybe for diseases like IgA, the way that we're thinking about it is that probably what we need to do is not just profile the kidney, but also profile other tissues like blood, and maybe ultimately, ideally, perhaps some mucosal associated lymphoid tissue, because as you know, say for IgA, patients get flares of disease often when they get a pharyngitis, for example. And so one of the questions has been, well, do the antibodies originate at the mucosal surface? So one of the things I didn't show you here is that we're able to do single cell antigen receptor sequencing, so BCR and TCR-seq. So you can see how we might be able to compare the BCRs in blood and the BCRs in kidney, say for IgA nephropathy, to say, well, is there any commonality? And if we compare controls and disease, what are the features of the clones that are perhaps producing pathological antibodies? Similarly for FSGS, I guess with FSGS, probably it's not so useful necessarily for that, because the thing that gives patients with a transplant FSGS is some soluble factor, soluble circulating factor that they have that isn't likely to be cellular, right? That when it perfuses the kidney, it then causes disease again. So I think probably one would have to then it would be more useful to do that on the patient's own kidney to see if there was anything kidney intrinsic rather than circulating to approach the FSGS.
Okay, thank you. The next question is from Sarah Aitken, who asked two questions. I think you've already answered her second question, so we'll just focus on her first question. She says, we often see a lot of immune cells, including neutrophils, as an artifact of surgical handling. Do you see this in the samples that you have from the donor kidneys? Could this be contributing to the innate response that you are seeing in the adult kidneys versus the embryonic fetal samples?
Yeah, so I guess it comes to the question of, well, there are two things in this, right? It comes to the question of what is artifact? So in fact, we know that if the kidney is perturbed in any way, and we best know this from mouse because we can just manipulate it, and it's much more difficult with human. But if there is damage to any part of the kidney, and that might occur physiologically during life, if, for example, you get a bit of a low blood pressure, if a tubule becomes unhappy, or if you deposit some uric acid stone. So if there's release of damage associated molecular patterns, there will be neutrophil recruitment ultimately, via inflammasome activation, IL-1 beta generation. So I think part of that may well be a physiological response, because with our pathologists, sometimes if we have severe ATN post-transplant, right, when we do our day 7 biopsy, because they've got delayed graft function, sometimes the pathologist will say, well, there's a whole bunch of neutrophils here, but we ignore that because they've got severe ATN, and I think we can perhaps forget that an innate immune response is an important part of the response to kidney damage. We maybe pathologically we focus a lot more on adaptive immunity. Anyway, in terms of this specific question, no doubt that surgical handling, can do that, but the embryonic kidneys, they are also surgically handled pretty robustly, and at that stage there are certainly neutrophils in the fetal kidneys, and if anything, the way that those were handled might be more aggressive than the adult kidneys. So particularly, for example, the tumor nephrectomies, but also even the kidney transplants where they are harvested with a view to transplanting that kidney, so they're pretty evenly handled. But I would say I don't think that will explain the difference between the fetal and adult, because the fetal ones are also prone to just as much or perhaps even more trauma.
Okay, thank you. So the next question is from John Tadros. He says, you've clearly demonstrated single-cell transcriptomics ability to generate a reference human kidney atlas. Given so much renal pathology is affected by proteins, antibodies, immune complexes, do you think that single-cell transcriptomics will provide sufficient information to infer pathology mediated by proteins, or do you need new technologies, single-cell spatial proteomics?
Yeah, so I think that's a really good question from John. So I guess that comes to what's the disconnect between the transcriptome and the proteome. And so we know that for some, and we have done one study by bulk RNA-seq, where we did bulk RNA-seq and mass spec proteomics on cortex, paired cortex and medulla samples from 5 kidneys. And actually we found that overall but I should say that, whereas you can get information on 20,000 genes, you can only get information even by mass spec on 3,000 proteins. So it's much more difficult. And we actually found that for most transcripts, that the correlation with the protein level was pretty good, but John is right, there are some that really are not similar, and so I think that ultimately he's correct that we'll need to use this as potentially as a combined approach, and there are ways of increasing the protein spatial information. So one of them is mass cytometry, right, where you use heavy metal labeled antibodies. So then you can get information on 50 plus markers, because you're not limited by the confines of the light spectrum. Or the other thing we've been trying is iterative strip and staining. So this is where you put on your 4 antibodies, you take your image and get your 4 colors, and then strip off, apply a second set, and we've got up to about 35 markers using that. And so I think it'll maybe ultimately be, combined approaches like that.
Okay, thank you. So the next question is from Louisa Moore, who has 2 questions. Do you have any plans to introduce single-cell genomics into this work to better characterize the resident immune cells in normal kidneys? And given the audience, how can pathologists contribute to this kind of work in the most meaningful way?
Yeah, so single-cell genomics, yeah, I mean, so one of the things that we've been doing in fact with some of Louisa's colleagues at Sanger is starting to dissect different parts of the kidney that potentially we can then look at the sequence of the cells within the dissected bit, as well as their transcriptome. So that sort of starts to get you to that, and starts to get to the very interesting work about, for example, how cells accumulate somatic mutation with life and whether that might contribute to diseases like chronic kidney disease with aging. And that's one thing we're really interested in. And to the second part, how can pathologists get involved? I mean, so as a nephrologist, I have a really close relationship with our nephropathologists. We first of all, we completely rely on them for diagnosis. Going down there and sitting at the microscope and looking at different features is incredibly helpful. And utilizing, for me to come at, say, different cell types and whether something is normal or abnormal in our single cell or spatial data, it would be foolish to do that without the expert knowledge of pathologists that have looked at this over many years. So I think it's critical to have pathologists involved in us trying to interpret this new data. But I think also ultimately, I'm really keen, and in fact, I have a trainee pathologist who's interested in renal pathology, who is part of the lab, and what James McCaffrey has done is to begin to put through the ethics for us to use excess clinical samples to apply these technologies to. Because I think for us to take this to the clinic, we need trainee pathologists that are coming through to embrace these technologies, and for us to have the capability, the pipelines, the ethics, etc., to be able to apply these technologies to clinical samples. So I think really getting trainee pathologists on board with labs that are doing this sort of work. And as I say, we have. I'm really delighted that we've got James involved on our side.
Thank you for that. And it's probably worth noting that James McAfee is partly funded by the GSPS grant schemes as well, which is a very good link for us. And given that we're now reached 3 minutes past 10, it's a good time to stop. We have a tradition of not going too far over time with these talks. So can I just end by thanking Menna again for a really fantastic and inspiring talk. Thank you very much indeed, Menna.
Thanks very much all. Have a good day. Thank you.
Thank you and goodbye.